let me recreate. Okay.
Got it.
No.
Okay, so that's great. Okay, let's get started. Good morning, good afternoon, and good evening, everyone. Welcome to our workshop. My name is Jen Chang, and from UC Berkeley, I'm your host today. And our guest speaker is Scott Cain. Before we start it, and I would like to give an acknowledgement to the organizers. They are Ming Yuan from Texas A&M, Xiaohui from Caltech, Mingjie from Duke University, and Chen from UC Berkeley. This workshop is supported by NAASC and co-organized by the Plant Postdocs. Scott is a research professor at Penn State with over 25 years of experience in bioinformatics, specializing in genome database development and genetic data visualization. He managed the generic model organism database project, which produced a widely used tool like JBrowse and TripPile software that many of you may have used, including the genome database for RoseACI and NoPulse. He also held senior roles at Wormbase and the Aliens of Genome Resource and Lab project, including Virus-seq and the Human Cancer Model Initiative. He received his PhD degree from UC San Diego and has spent his career building the infrastructure that make biological data accessible to researchers worldwide. For the past 2 years, Scott has been part of the Galaxy project, a free web-based platform for biological data analysis, where he feels very at home because Galaxy shared his commitment to making scientific computing freely available to everyone, regardless of their computational background. With that, let's welcome Scott.
All right, great. Thanks. Everybody can hear me, I hope. And that should have shared my desktop. And now you should be able to see my slides. Okay, terrific. Right. So thanks for that introduction. Yeah, so I'm at Penn State. Here's my contact info, my email address, and this is my handle on Mastodon for anybody who uses Mastodon. That's kind of the only social media I really kind of care for these days. Okay, so I'm going to try to cover four broad topics: introduction to Galaxy, kind of how Galaxy is used, which hopefully will include a little bit of hands-on stuff, and then we'll talk a little bit about scientific applications, and I'll finish up with some AI and machine learning stuff in Galaxy because it's cool and fun. Okay, so what is Galaxy? It is multiple things. It is most fundamentally, I guess, software that combines tools and workflows and computation and data to let you do all sorts of primarily bioinformatics analysis, although there are other types of analyses you can do as well. It is also a very large community that works on training and publications and collaborations with other things, and of course web interface, which is also part of the software. So try to make sense of some of that. So the Galaxy project has been around for just over 20 years. We celebrated our 20th anniversary last year. Right now, there are over 10,000 tools that are available in Galaxy. There have been, since the inception of the project, since we've started tracking these things, I don't know when that started, but over 900,000 workflow runs, 350 million total data sets. So a data set is any sort of data that you've either uploaded or that's a result of a computation. So that's a lot. We've run over 195 million jobs. There's over 690,000 registered users. This one was painful for me because it's really close to 700,000, and I really wanted to put 700,000 there. That's not quite there yet, but soon. And there have been over 22,000 citations of the Galaxy project in publications. So like I said, there are tools and data for every users. There's over 100,000 biocontainers, which are software wrapping tools covering pretty much the majority of bioinformatics tools. Basically, any open source bioinformatics tool that you can think of is probably in Galaxy already. In addition to that, there are pre-installed reference genomes for lots of reference genomes, including lots of plant ones, Arabidopsis, rice, maize, anything that you would expect to be there is certainly going to be there. And if you look for one and you don't find it, let us know, because it may just mean, like, this actually happened to me when I was getting ready for this talk. I was like, why is Arabidopsis not in our reference data? And it was there. It just, there was one index that needed to be rebuilt. So you just have to, if something like that happens, give us a poke. We'll find it. You can also run interactive analyses. So it's not just kind of like click button, run this job. You can also do things like write Jupyter notebooks that you can run Python or RStudio in to do interactive things, to do more more detailed sorts of things. You can we have access to our in the back end. We have access to lots of GPUs. So if you're using high memory or GPU based software, you can do that through Galaxy. And you have histories that track all of your jobs, so that anything you do, you can go back later and say, what did I do there? Or you can share it with other people and you can show them what you did. And so that is really great for reproducibility as well as collaboration. And there's all sorts of new data and technologies. There's single cell stuff. We can do imaging, and it's a lot of really amazing technology that is access to you for free. I forgot to make a slide. I was going to make a slide at the very beginning that says, why should you use this? And one of the reasons is because it's free. There's high-powered, high-performance computing that you get for free, which is amazing. Okay. Okay. So the real work that can be done in Galaxy is taking individual steps of analysis where you run this tool, get a result, run another tool. You can package those up into workflows so that if you're doing the same sort of analysis over and over again, you can just say, okay, here's a new set of data, run this whole workflow that I've been doing before and do it again on this new data set. And so you can build them from scratch. You can get them from elsewhere. There are a couple of different places you can get Galaxy workflows. And so that's really very powerful. You can also, if you're interested, you can do Galaxy on the command line. There are a couple of different ways to access it, either via a tool called Planemo, which is a command line tool where you can basically tell it, can script running things inside of Galaxy. So you still have access to the high compute via scripting. So Planemo, or there's a Python package called BioBlend that knows how to talk to the Galaxy API. So that so you can do that, and basically against any public Galaxy instance, you could do that. Or if you have one installed internally, like at your institution, a lot of universities have their own Galaxies installed. And yeah, it's for everyone. So I'd say kind of this middle one is probably not of interest to most people in this group, but on the outsides, you've got analysts and researchers where you've got, again, lots of data, lots of tools, lots of workflows that basically at a push of a button, you get access to free compute. And then on the other side, students and educators, you have the ability to, there's lots of training. I'll talk a little bit about training options in a little bit, but there's also global support literally around the world. There's basically always somebody awake who knows something about Google or that knows something about Galaxy, excuse me. And the homepage is galaxyproject.org. I'll have a set of links at the end to share with everybody. So that's probably the first one I mentioned there. Galaxyproject.org is our homepage. There's also galaxies all over the place. The one I'm going to talk about mostly today is usegalaxy.org. That is housed at the Texas Advanced Computer Center at the University of Texas in Austin. But there are lots of other ones. The other two kind of top level, I guess, the really big ones are usegalaxy.eu, which is in Germany at the University of Freiburg, and usegalaxy.org.au, which is at the University of Melbourne in Australia. There are also lots of kind of country level use galaxies. So there's like usegalaxy.fr in France, usegalaxy.be in Belgium. There's several of those. So anyway, and there's a map of instances down here. But basically, So usegalaxy.org, again, has a lot of compute power. So does usegalaxy.eu and usegalaxy.org.au. And those country level ones may give preference to people who are from those countries. And so you might get even more high powered, but not as easily to access for the wide public at those at the like country level use instances. Okay. So at usegalaxy.org, this is the usage data for the last year. We have a monthly of over 4,000 new users every month, monthly average. Like I said, at usegalaxy.org, we've got over 460,000 total users every month. There's about 75 million jobs that are run at usegalaxy.org. So it gets a lot of use. And like I said, it's powered by both the Texas Advanced Computer Center and Jetstream 2, which is at the University of Indiana. Both are publicly funded via NIH and NSF, which is the same as Galaxy, also funded by NSF and NIH. Okay. So terrific. So that's kind of a background, gives you a little bit of idea of what it is and why you might want to use it. If you want to do hands-on in a little bit, now might be a good idea. Now might be a good time to create a Galaxy account if you don't already have one. It's really easy. You go to usegalaxy.org, you hit login or register, and then you hit, create an account, fill in your information, and hit create. Boom, you're created. What happens when you do that is you automatically get 250 gigabytes of free storage space to upload your data to. In addition to that 250 gigabytes of storage space that comes for free, you also get kind of temporary scratch storage space that sticks around for a month and then gets automatically deleted. You get a terabyte of that. In addition to that, if you have some sort of cloud storage technology like Dropbox or an Amazon Web Services S3 bucket, you can also attach that and use that as storage. And if you really need it, don't tell anybody I told you, but if you ask them for more permanent storage, you usually will just get a terabyte right away. So don't tell anybody I told you that though. Okay. And that sticks around for a very long time. I don't know how long, but many years. I can tell you that before I started working at the Galaxy project, I used Galaxy for things. And so when I came to Galaxy and started working, I logged into my account that I hadn't used in a while. And there was an analysis that I had done 12 or 13 years before, and all the data was still there. So it sticks around for a while. Okay. Typically, what you do in kind of a typical analysis in Galaxy is you load or pick your data sets. If there's some preloaded, or you upload something from your computer, or you get it from some other web-based data source. So you get some data, you pick a tool to run on it. Something that makes sense, whatever, and then you hit go and you check the outputs to see if they make sense. And somewhere down the line, hopefully you'll publish and share those analyses. But certainly Galaxy can be used to create publication-ready figures and lots of visualization tools. Also, publishing reproducible workflows is a really great way to kind of supplement like materials and methods sections for sure. And I'll talk more about that when I get to the AI stuff, because there's cool stuff you can do with that. And you publish all the data that is kind of that you've created as a result of doing your analysis. But typically what happens is you get to this step 3 and you're like, well, I need to do more. That's pretty much what everybody needs to do. You run a tool and then you're like, okay, now I need to do more. And so you go back to steps 1 and 2 and you run some more analyses. And basically your history keeps growing while you do that. And so for a given analysis set, you can just keep stacking tool output results on tool output results. Okay. Before I get to the histories, though, again, using loading user data, like I said, there's lots of ways to do it. You can upload it from your computer. You can fetch it from, if you have a URL that points at, any of a variety of potential web data sets, you can do that. Again, if you have something like Dropbox or what is it, Google Drive or AWS S3 buckets, that sort of thing. You can just mount those as drives and get data there. You can do it. There's a command line tool as well. There are also several tools that which we'll use later on today on the hands-on part, where you can say, oh, I've got this SRA accession or this list of SRA accessions. Um, uh, I have do not disturb on my phone, but the one person who gets to get through that just called and made my phone ring. Anyway, sorry about that. So you can give a list of like SRA accessions and it will just go fetch them. So that there's a lot of ways. So that kind of comes under all these import options down here, like from EBI, NCBI, UCSC. Okay. There's also lots of reference data that is included. Like I said, lots of reference genomes, indexes built so that they can be used with a variety of tools. There's also data sets that are kind of large shared data sets in a variety of fields, and you can get those from Galaxy as well and access them without having to kind of load them in yourself, which is really nice. This is kind of not kind of, this is exactly what the Galaxy interface will look like when you're using it. There's different parts. There's basically on the very left-hand side, there's a thing we call the activity bar, which basically is quick access to a lot of commonly used features like uploading or searching for tools or looking at workflows are available, what workflows you've run, that sort of thing, switching to different histories. That's what's there. And then there's this panel that is right next to it. Frequently, that's the tool panel, because that's usually very frequently what you're doing is selecting from a large set of tools. But if you click on something else, it might be a workflow panel or a history panel or something like that. But that's basically where you get the the tool to work with that you've clicked on from the activity panel. Okay, then next to that, kind of in the center, is the data and the select panels. This is where you're doing the bulk of your work. You're either configuring to run some analysis and then hitting go, or you're looking at the output of some analysis. And then all the way on the right then is the history of everything you've done in this analysis that you're working on, right? And so you can have multiple histories that keep track of things. My advice to you, when you create a history, give it a name so that it makes sense, because it will give you Untitled History 1, Untitled History 2, and 2 months from now, you will not remember what those are. Okay. Anyway, so when you pick a tool that you want to launch, for instance, here is FastQC, which is a good tool for analyzing your sequencing results to see if the data are quality sequence. So it does quality control. So this is what you would do. You would click on FastQC, and then you would get something that looks like this, where you can say, okay, well, I want to run it on this thing that's already in my history, and Galaxy will place in this the thing that makes sense. Now, there's only one thing in your history, so that's there's not much for it to do to figure out. But if you've got hundreds of things, it'll say, oh, well, the only thing that makes sense to run on this tool is something that's like a FastQ file. So it will look through your history and say, pick one of these FastQ files to do this. And it does that for virtually any tool you run to give you to make it easier for you to figure out what the right thing to do is. So once you've picked that thing, then you can hit the Run Tool button, and there we go. And oh yeah, so the data's already there. Yeah, and then you select the topic. Okay, so I had things for this, and then you set the options, and then you hit Run the tool, and then that gets submitted. It basically you'll immediately get a green box in the middle that tells you what was submitted. And it'll also tell you what outputs you should expect, because tools can produce multiple outputs. In this case, it produces 2 potential outputs, and then it puts that into the history. Initially, it will be gray, which means it's waiting in a queue to be assigned somewhere to run, and then it will turn orange when it's running. So in this, this is what it looks like when it's running, and then when it's done running, it will either be red or green, and hopefully all your jobs are green. So red means it errored in some way, so you might have to dig out what happened. Okay. All right. So then after running, you can say, okay, show me what it looks like. There's this little thing here. It's kind of hard to tell, I am sure, on most screens. It's actually kind of like an eyeball that's in kind of like a little frame. So frequently I'll talk about poking the eye. So if you poke the eye, you get to see what the result looks like. So this is an output result from FastQC showing the sequence data is pretty good. So that's nice. So then you can say, okay, well, I've got good sequence. Now I'm going to do something with it. So I'm going to run Minimap2 to align it to a genome. So again, you look for Minimap and it will say, okay, you can map something with Minimap2, and then it will open up this thing in the middle. And so again, it will look and say, okay, it's not going to pick the last thing you created because the last thing you created doesn't make sense for running Minimap, but it does see that, again, the first item in the history is a FASTQ file. So it will put that FASTQ file in there. It's the only thing in this history that makes sense in this context. So it will put it in there for you. And then you have to tell it, okay, well, what do I want to align it against? And so in this example, it's aligning it against human. But again, there's a lot of reference genomes here to pick from. And then after you do that, you can just hit run again, and then off it goes. And again, gives you this little green box that says it's been submitted, and you can expect one output, which is a BAM file after running Minimap. Okay. So again, doing these things step by step makes perfect sense when you're figuring out what you want to do, or you want to do something relatively short. But if you're doing something big, it often makes sense to make some sort of workflow. So this is an example of what a workflow looks like. It is zoomed way out so that you can see the whole thing. It's quite big. There's lots of steps. This is to do scaffolding, creating DNA assemblies using Hi-C data. And so there are a lot of steps here, and it will presumably run quite a while. But you can do a full-blown genome assembly in Galaxy. So that's great. So you can build this as a custom workflow, or in the example before where we generated a history, there's a history menu that's one of the options in the history menu is extract this into a workflow, and then it'll create a workflow for you. Or you can get it somewhere else, like at this website, which is iwc.galaxyproject.org. IWC stands for the Intergalactic Workflow Commission, which is maybe slightly silly in terms of names. But basically, this is a website that has over 100 curated workflows that you can just go read, make sure it does what you want it to do, works on the type of data you have, and then you can just say, yeah, put this into my Galaxy so that I can run this workflow on this data. Once you've done some analysis, you'll frequently want to figure out what the data looks like. So there's multiple visualization and ways to create interactive tools, charts, graphs, genome browsers. The IGV is integrated into Galaxy. So if you have data that makes sense to view in a genome browser, you can just put it straight into IGV inside of Galaxy, which is really nice as well. In addition to that, you can also write Jupyter notebooks so that you can have Python or R that does exactly what you want to do with, starting with a data set that's in your history and then producing something as a result, either another data set or creating a graph kind of like this. This is an example of a Jupyter notebook that has Python in it that that took some data and then plotted it, did some math and then plotted the results. So it's really nice. I mentioned training before. So there is a website called the Galaxy Training Network. It's at training.galaxyproject.org. There have been over 500 people who've contributed to it and over 500 tutorials that have been created. Right, let's see, right there, that little flower there, that's me. So I am on this list as well, although you'd see that there's about 500 contributors and about 500 tutorials. Maybe there's a one-to-one correspondence. There definitely is not, because I've never written a tutorial from start to finish, but I've edited several of them. So that's kind of the way it works. Somebody writes something, somebody edits it, it's great. Anyway, but there are 35 different topics which go from kind of Galaxy starting, how to start. In fact, that's what's on this. This is kind of the start page of the Galaxy Training Network. page and start here. This is introduction stuff, how to do things. And then down here, there's like lots of different fields that are options. Cutoff just below the cutoff at the bottom of the screen here, there is a plant topic. But I'll show you that in a minute. So that's again, over 500 tutorials. There is so many topics: transcriptomics, genome assembly, single cell, lots of things, so many things. Okay. And very conveniently, in addition to that, this is going to be our third annual event in May called the Galaxy Training Academy, which is an online week of training. There will be TAs online 24 hours a day for the whole week, May 18th through 22nd, because basically we've got people in North America, we've got people in Europe, and we've got people in Australia. So basically, there's always somebody online in the Slack, answering questions and helping people. Last year, we had over 3,000 participants, and there are multiple training paths, that you can, whatever topic you want to learn something about and go into detail. This is the short URL for it. I'll have that at the end of the talk as well. So anyway, so I encourage you, if this is something that's of interest to you, free computation, this is a really good way to get started. So that's in just under a month. Okay. In addition to that, there is on galaxyproject.org, there's also an events page where you can go and see what things are happening that might be of interest to you. A variety of topics, webinars, meetings, those sorts of things. So that's useful as well. Okay. Now I'm going to talk a little bit about scientific applications. Like I said, there have been over 22,000 citations. I didn't check the number recently. We hit 22,000, I think, towards the end of last year. So maybe at 22.5 now, maybe. I don't know. I haven't looked. But it is a wide variety of topics. In fact, I was just talking to somebody this morning. They found an anthropology paper where they had used Galaxy to do some analysis. So whatever science you're doing, you could probably use Galaxy to do it. Whatever. Okay, let's see. And then this is I borrowed this slide from somebody, but it's pretty neat. It's basically lots of different types of topics you can cover, including comparative genomics. This VGP project, obviously, it's not plants; it's vertebrate genomes, but I think there are going to be similar projects to this kicking off in the near future, where they're just using an enormous amount of compute. But what their goal is to sequence every vertebrate that is known. So there's like 70-ish thousand of them. So there's going to be a lot of new genomes coming along down the pike from them. But I think there are going to be very similar projects in plants as well, although they probably already exist, excuse me. But yeah, single cell, proteomics, machine learning, so many topics. Yeah. Okay. So for plant science at Galaxy, like I said, at the training.galaxyproject.org, there is a plants topic where you can do tutorials on how to do assembly. The chloroplast genome assembly is relatively easy, kind of low. It doesn't require a lot of compute, but you can do, like I said, you can do full-on genome assembly as well. After you've got your genome assembled, you can do genome annotation. There's a tutorial on that. You can do transcriptomics. There's a tutorial on how to do that. Let's see. You can do transcriptomics type things, single cell, doing analyzing fair data, workflows, and research. There's a tutorial on that, and also microbiome associated with plants as well. I should mention that, so while I probably am obviously not a plant biologist, there are a couple of plant biologists in the European Use Galaxy group. So they are the ones who've written most of these tutorials. And I also asked them for suggestions on things that are worthwhile to talk about. So anyway, like I said, there's a lot of plant stuff already at usegalaxy.org. So that's great. There's also kind of several generic tools for doing image analysis. Like this one is again, I don't know that much about image analysis. There's this really nice YouTube video that kind of it actually has really high production value. I'm very impressed with the people who made it. But it's basically this sort of thing where you can do image analysis, and it doesn't really matter what the underlying thing is that you want to do the analysis of. It can be cells, it can be plant growth, taken from overhead, or astronomical objects, or things underwater. But it can do classifying. You just have to train it, and so you can do image analysis like that as well. Let's see. So we've gotten to the point where I'd like to do hands-on stuff. What I'm going to do is exit out of this slide deck, copy these things. These are the things that we'll need as inputs, and I'm going to put those in the I'm going to put those. No. Oh, no, this is this is the one I want. Okay. Good, very good. I'm going to grab these and put them into the chat, I think, as long as I can. We'll see what happens. All right. How did I lose it? Huh. Am I still connected?
Hi, Scott. Yes, you're still connected. We can still hear.
That's good.
That's
I'm really
good.
glad to
Yeah,
hear
you're
that.
connected for the whole time. Yeah, we're seeing some terminals. I believe you just want to show a
Yeah.
browser.
Yeah, that's
Yeah.
let's see. Oh, I need this. This is what I need. No, here we go. Here we go. There we go. I put into chat these links. That's what I was looking for. I couldn't find the silly chat window. Okay. Okay, so this is what a workflow looks like after it's been running for a while and then had an error in the step. I was running this morning. So I guess we'll
Excuse
see what
me.
happens. Yes.
Sorry, we don't have it in the chat.
Let me come back and look.
I'll help to put the link in the chat.
Oh, you know what?
Yeah, is that okay?
I didn't hit return.
Oh.
There we
Okay,
go.
great.
How's that? Okay. Okay. So basically what we're going to do is I'm gonna let's see. I'm gonna open a new page. Okay, so this is what Galaxy will usually look like when you start it, except for the stuff on the left. I'm going to switch to this history. There we go. And this one has all of the files that I basically was saying we needed to start, but I'll still show you how I got these in. So basically, the first thing I did was I went to Tools, and right underneath that is Get Data. So I'm going to get some data. And the second thing in here is downloaded extract reads from FASTQ format from SRA. Okay, so I will click on that. Right. And then, like I said, this is kind of where everything happens. And so it says select the input type. And yeah, we want to give an SRA accession. Or you could do a list of accessions. So that sort of thing. But if you do accession here, you can actually put multiple things in here separated by commas or spaces. So let me get the chat that has the accession I'm going to use. This is basically a RNA-seq experiment on Arabidopsis where they did RNA-seq on the roots. For some conditions, I only got one of the accessions because I wanted to kind of limit how much we were doing. But let's see. So so I put the accession in there, and there's not really anything else that's needed to be done here. the defaults are fine. You can do things like have it email you when it's done running. So if you're doing something that's going to run a very long time, it's actually really nice to have that. I generally don't. I just check back. I am not going to hit run tool here because, like I said, I've already got this data here. But you would click run tool. Okay. And then for these other two things, this is I actually put the Arabidopsis genome in here, both because it's fairly small, so it's not that big a deal. And I wanted to make sure that the names for chromosomes that were used in my GTF file are the same as the names that are in the FASTA file, because naming things is one of the things that's really annoyingly difficult about bioinformatics. So basically, I did that to make sure they were named the same. So to get these uploaded, I click on the upload button, and here, there's lots of options here, but the one we want is the paste and fetch. So basically, I paste in URLs and then I hit go, and that will then it will go fetch those things. So the URLs I need are the two that are in there. One for the Arabidopsis genome, copy that, put it in here, and the one for the GTF file that has all the gene annotations in it, copy that, put it in here. And then I would hit start, and then it would immediately, I think it immediately turns green unless I mess something up, and it would turn red. But again, not going to do that because I've already got the data in here. And then I can close this, and it will continue to load while if it won't all load while you're while that dialog is open. You can close it though, and it will continue to load in the history. So basically, we've got these four things. We've got paired end sequencing data from SRI, and then the two files that I provided, the GTF and the FASTA of the Arabidopsis genome. Okay, so what we want to do then is the other URL is in there is the iwc.galaxyproject.org URL. It goes to this page. Well, it doesn't go to this page. It goes to specifically to a workflow page. In fact, why don't I just do that to make sure I get the right one instead of just assuming I'll remember. Okay. Okay, so this is a variant calling workflow that uses paired end data. So we can do, I don't think that the SRA data that I got was specifically for variant calling, but we can certainly use it for variant calling because they made the data public, so we can do with it whatever we want. And this one is also ploidy aware, which is also really, really useful, particularly in plants where you've got crazy ploidy. So it basically gives you a description of what it's going to do. It gives you a list of things that you have to provide as inputs, which is nice. And then it tells you what it will give you as outputs. And then at the bottom over on the right-hand side, there's this tool that says, run this workflow somewhere. And it gives you an option of selecting where do I want it to run. So there's all these options, but I'm going to pick .org because that's where I have an account. Although, well, I have accounts at EU and AU as well, but .org is where I want to run. And then down here, there's actually two buttons that are also really nice too. So there's try with example data, which is really very useful when you're trying to learn what something does. Basically, all of these workflows have sample data that just come with them, so you can just load it into your account, run it, see what it does, see if that makes sense to you, and that's great. Or I can just hit the launch workflow, and it will take me to a window that looks like this. Well, not quite like this. Come on. There we go. So it's basically it loads the workflow into the history that I was already using. So these are the four things that I already loaded. Then it loads the data. I'm guessing it's a little slow because hopefully a bunch of other people are using it too. So that's maybe why it's a little slow right now. Okay, and like I said, it basically figures out on its own what input data you're supposed to be using. So the things you have to provide is a paired collection. It's basically forward and reverse reads. And so it's like, okay, I've already got one of those. So offers it to me. This is a drop down, but since there's only one thing that fits this category, there's only one thing in it. And then it says you have to give a reference genome. So again, there's only one of those in here. So I did that. If I hadn't provided it, this would instead be orange. There'd be a red exclamation point here, and I could hit upload and have kind of a similar interface where I could upload the data from here as well. But I don't need to do that. And then you have to set the ploidy. And since we're working with Arabidopsis, that's a simple 2. And then you also have to provide it with a set of annotations for genes in Arabidopsis, and we provided that as well. Okay. Other things, let's see. You can edit the workflow if you want. We're not going to do that. It will show you, oh, that's going to be too small. I'm not even going to bother. Okay, I'll show it to you in a minute. There's also there's help up here at the top. There's also settings that you might want to use. A few of them are kind of useful. I'm not going to use it right now, but this one is send outputs and intermediate to different storage. So basically the idea here is that this is the workflow that has multiple steps. Most of the steps, you don't care about the outputs except as inputs for the next step. So if you set this, if I set this, it will send the intermediate steps to that kind of scratch temporary storage that I talked about that gets automatically deleted after 30 days. Because if all I care about is what's the end result, then I don't care if it gets deleted in 30 days. I still have my result. So that's nice. But I'm not going to turn that on right now. But that's really useful. Again, there's this notify me when it's complete. This reuse jobs with identical parameters is really useful if a job that you're doing, if a workflow that you're doing errored at some point, you did something to fix the error, and then you can say rerun. And all of the jobs that were successful before, it'll just reuse those. It won't redo the calculation. It'll just say, oh, that job's already done. I don't have to do anything. And then the last one that is also useful, it's the top of the list, but this one that is also useful is send results to a new history. I can't tell you how many times I have done something like this where I am like at IWC or another tool that will take me into Galaxy. And it'll take me to a history that is completely unrelated. So I can just say, oh, this is not really where I want to be running this. So send the results, everything gets run in a new history. Then I can deal with that later. Okay. So anyway, so this is ready to go. I am going to go ahead and hit run workflow. And we'll see what it looks like when it goes. Okay. So initially what happens when you hit run workflow, it basically creates all of these steps that get scheduled. It will show you a graph down here of what's going to happen. It's usually very small because workflows have a lot of steps. And again, it's kind of slow, probably because a bunch of people are using it. And that's okay. Okay, so yeah, so this is kind of what it looks like. The things that are green are things that are already, are done. These are the inputs, actually. I can zoom in so you can take a look, so we can show you what that looks like for real. Yeah, so these are the inputs to the various steps. And then all of the things right now are gray. If I come down here, there's basically all of these things were scheduled. Oh, this one has started running. That's building the database for the snip effect caller. So that's easy, but it's also a very fast step. So that'll probably complete pretty quickly. These other steps, okay, so the FastP is now running as well. This will take a long time. But anyway, this is what it looks like when you start a new workflow and things start happening. And so what will happen is it will just march across the screen here until it gets to the end, or until it encounters an error. So if I switch right now so there's like kind of these control buttons up here. This is create a new history. This is switch to a different history. And then this is history options. Since I'm here, I'll point them out to you. Things that are really useful at the bottom is share and manage access. You can share histories either with just collaborators, or you can share them with the world. So for instance, if you want to include it in a publication, you can share it with the world. Which is also useful combined with archive history. If you don't want any changes to a history because you've published it to a paper, you can archive it, and then it'll basically be read only. You won't be able to touch it anymore, but people will still be able to see that history and create copies of it, which is really useful. Okay, but what I'm going to do is switch to this one just so that I promise I didn't do this on purpose, but yeah, the second to last step here errored. And it's really useful sometimes to see what happens when things error. So I would click this to basically see what happened. There's nothing there that is showing you things, but if I click on this, this expands, this history element expands to kind of, it gives me the error option, it gives me some information about where the error came from, but there's a little bug right here, and I can click on this, or I could have clicked on, it was here too. So basically it tells me, let's see, error parsing. See, that's something I don't know if I can do anything about that. When I see an error like this, it makes me think something happened that was weird. That all of the steps before it completed without an error. They're all green. And so what I would be inclined to do at this point is try rerunning without doing anything. Because sometimes, when you're working with a compute cluster, sometimes one of those computers is finicky and throws errors for no reason. So kind of the first thing I might do is I might just try running it again to see what happens. So to do that, I would come back to the work. I click on this button here, workflow invocations. And that's the one that I just started 4 minutes ago. I don't care about this one. This is the one that I started 3 hours ago. So I'm going to switch to that and then see how everything is green, like I said. But then if I move over here, the second to the last step, this one is red. So what I'm going to do is I'm going to hit rerun. And so what it's going to do is basically look at the workflow that I already ran. It says, okay, everything here is the same. It just wants to rerun the same thing, but I'm going to hit the reuse jobs with identical parameters, and I'm going to rerun the workflow and hope that, sometimes so it'll take a little while to figure out, okay, these things are all done. And then we'll get to this step and hopefully it will run without error. We'll see. Well, we probably won't see because we're running low on time, but we'll see what happens. I'll have to let everybody know what happens, I guess. So anyway, so that is using Galaxy. What I'd like to do at this point then is, is basically running through very quickly, figuring out that it has all of these things already. So that's good. What I'm going to do is switch back to my slides and kind of wrap up what else I've been doing. So basically, these are just screenshots of things that I've already talked about, but they're here. Okay. Oh, this one, the blank one, this was supposed to be the workflow when it was done, but I never got to that point. That's why it's blank. Okay. So I do want to talk a little bit about using AI and machine learning in Galaxy. There are several machine learning tools. There's a machine learning working group in the Galaxy community where the people get together on a regular basis and talk about things that they can do. This is a nice example of, it's not even bioinformatics, but it's basically taking handwritten numbers and using a machine learning tool to identify those numbers. So that's kind of a neat example. There is also a lot of work being done to integrate kind of chat type tools so that you can do a data analysis with natural language. There are things about using things like Claude and ChatGPT and Gemini that make me nervous, but there are a lot of things that make me nervous in general in the world as a whole. But in terms of Galaxy, I'm not too worried about it because anything you do in Galaxy, because it's part of a history, you can always go back and figure out what happened, even if you're letting something else make those things happen. So I'm a little less worried about it, but it's still check your outputs. But there are tools being worked on and some already released for doing things like chat and having that interact with your content in in Galaxy. So specifically, there's something called Galaxy MCP. So MCP servers are things that let you kind of interact with LLM tools via scripting. So basically, it's pretty amazing that so we've written a Galaxy MPC server that knows how to talk to Galaxy. So you can say, you can type into whatever tool you're using, like Claude or ChatGPT, talk to my Galaxy instance, and figure out how many data sets there are, and if there are any that are for this genome, and if there are some for this genome, what analysis did I do? Summarize it, and suggest next steps, that sort of thing. So basically, the way this works is you have the user right here type something into ChatGPT or Claude or whatever, and then the MPC client that is inside of Claude or ChatGPT knows how to convert that to code that can talk to the MPC server that's running for Galaxy. That's what happens here, and then it goes to the Galaxy MPC server that then knows how to talk to Galaxy, or actually the BioBlend API that I talked about before that knows Python, and then that talks to your Galaxy instance. And so this is really pretty powerful. Current downsides is that, well, I don't have it on, I think it's on the next slide, is that this MPC server you have to run right now yourself, but you can have Claude or ChatGPT or whatever you're using, you can have it build the MPCs and run the MPC server for you. So you don't actually have to know how to do that. So things that it can do, like I said, it can connect, figure out information about the server. It can look for tools. You can ask questions like, I have this kind of data. What do I need? What is the most likely thing that I would use to analyze it? Are there downsides to using that thing? So it knows how to interrogate the tools that are used, because the tools that are in Galaxy are all very well documented. They all have references. So there's a lot of information on each tool. So these LLM tools know how to talk to them and get that information out. Like I said, you can also have it interact with your data. Kind of the coolest demo that I saw using this tool, I don't have an MPC server running right now, so I can't show you, but I can tell you about it, is where you can have it say, okay, look at this history that I want to include in a paper. Write a materials and methods section that covers what was done in this history. And it does a pretty good job. You're always going to want to check it and edit it, but it does a pretty good job. And it's one of the things that is really good, again, because everything is there. There's not much for the LLM to make up, to hallucinate, which is not a great term. I don't really like using it that way. But because everything's there, it's just a matter of, basically, it's a nice formatter for you. And it can also interact with workflows. Like, it can go to that IWC website and get a workflow and import it in for you, and like kind of configure it and even run it for you if you want to let it. So it can do quite a bit. So there's like lots of examples of how you might want to do it. Like as a researcher, you could say, well, I have this RNA-seq data from these crop samples. Help me analyze the differential expression. So it will suggest tools, workflows, parameters, things like that. It can also explain errors. So I could have had to take a look at the error that I had on that step and say, what does this mean and why do you think it happened? I did not do that. I could go back and maybe try. But and for bioinformaticians too, it says, look for workflows and help me work. So there's a lot of tools you can use that will make this a lot easier for any level of bioinformatician or a person who needs to use bioinformatics. So things that exist now is this standalone MPC server that I said, you can have your chatbot install for you and run. There's a bunch of operations, 20 plus operations. There's called skills that it knows how to do. So and it works with any MCP compatible API, which includes Claude, ChatGPT, and Gemini. All of those work with it. There are other projects. A lot of these are for doing similar sorts of things. The one that has just been released in the last few weeks, I haven't had a chance, much of a chance to work with it, is Chat GXY. It has vision after it because I copied this from the coming soon to this already exists because it was just recently released and I forgot to delete that. So anyway, but it's basically built into the Galaxy interface right now, and it's context aware. So I should be able to go back to that page where the thing aired, and we can ask it what happened. Things that are coming are more high-powered things like a Pydantic AI agent that you can run in the command line, as well as a hosted MPC service, so that you don't have to run your own standalone MCP server. But that doesn't exist yet. But coming soon, I think. Okay, so AI, there's lots of things there. Both machine learning and LLMs are really cool, and it is pretty amazing what it allows you to do. I can tell you, I know I'm getting close to end. I can tell you that for a talk I went to at a fungal genomics conference last month, I used this the Galaxy MCP to write a Jupyter notebook that did some analysis that went beyond what was really easy to do in Galaxy. And it was really quite powerful. It required multiple iterations to get it right, but that's okay. I'm okay with that because it wrote code that I didn't really know how to do off the top of my head. I could have spent a few months figuring it out, but instead I spent a few days with an LLM and let it do it for me. So it's really powerful. These are basically our social media stuff. Like I said, I'm on Mastodon right now. But yeah, we have Blue Sky, we have LinkedIn. Also Element, it says Element, but that's really kind of more Matrix. If you guys know what Matrix is, it's a free distributed thing that's very much like Slack. And is annoying in different ways that Slack is annoying. Slack is also annoying, but Matrix is annoying in different ways. So anyway, but that's kind of the way we interact with a lot of people in the community. Okay, so to summarize, use Galaxy to accelerate your science. It does all kinds of things for you, including accessibility, making collaboration easier, making reproducibility easier, which is that is a difficult thing. You get speed advantages from the fact that yes, there's a very powerful compute clusters sitting behind it, but also letting you reuse things that other people have already developed makes life a lot easier. The connectivity, combining tools and visualizations all in one space, very powerful. It is very scalable. If you need to do large genome assemblies, there are ways to do that in Galaxy. And we're doing innovation all the time, like the AI stuff. And it also is also a really good way to deal with best practices for various sorts of analyses. So a lot of what is in those workflows that have been published and curated represent those kind of best practice thoughts. Okay. So much thanks to everybody on the US Galaxy team, and thanks to actually I should thank the European Galaxy team too, because they helped me as well. There's quite a bit of people. The community is fairly large. This is a screenshot from our last community meeting last summer at Cold Spring Harbor. This is the Galaxy and Bioconductor Community Conference. So I'm in here somewhere, but I don't really remember where. Definitely behind lots of people. So thank you. And here are a bunch of resources. This will go up on the website as well. There's, again, usegalaxy.org, EU, and org.au, but there's a lot more listed at this link. I didn't mention it, but at the European Galaxy site, they have a lot of kind of subdomains for specific science approaches. They do have a plants.usegalaxy.eu site that is kind of geared toward plant researchers. So you might find that useful. And I will point out that you don't have to be in the European Union to use usegalaxy.eu. You can be anywhere in the world and they will let you use it, which is true of usegalaxy.org and usegalaxy.org.au. Galaxy Project I mentioned is our homepage. Training.galaxyproject.org is the training network that I mentioned before. This is the link for the Training Academy that is coming up next month. This is the IWC website that I mentioned. I did not mention help.galaxyproject.org, but that is a Discord forum that is very heavily trafficked and is manned by several people in the project. Okay. I think that is probably my last slide. It is. Okay. So
Okay.
I'm going to see if the chat thing is in here. I don't think it is.
Okay.
I don't remember how to use it. Okay, that's okay.
Wonderful. Yeah. Thank you. Thank you,
All
Scott. This is
right.
a wonderful presentation. I believe all the audience have learned a lot new about the Galaxy. We have a lot of questions coming in, but our time is coming to the end. So what we'll do is we'll ask three very general questions that will be of general interest about the audience. And
Okay.
for the more advanced questions, people can feel free to reach out to Galaxy team directly. So the first question is about running the job. So do you have to stay logged onto Galaxy and keep your browser open while your analysis is
No,
running?
no. Absolutely
Yeah.
not. Because some of these things will run a long time. So like this job that aired out, I expected it to run probably a couple of hours, which is fairly reasonable. But there are other jobs that will run literally days. And yeah, you don't have to stay logged in. It will just continue to run. And then you can log in later and check and see how it's going.
Okay, sounds good. And another question about the Galaxy Training Academy. So one of the attendees saying that they have registered for the training, but they have not heard back if they were selected to participate or not. So
Oh,
what should they proceed?
there's no selecting to participate. If you registered, you can participate. Um, I assume that, boy, I don't know the answer to this off the top of my head, even though I just had a meeting with people about the GTA this morning, but I don't know the answer to this question off the top of my head. I assume that as it gets close, there will be like URLs and that sort of thing mailed out about like how to, there will be a Slack that is set up just for the GTA. And so I think the next thing you hear will be getting an email inviting you to that Slack and letting you log in. That will probably happen the week or so before the GTA. But yeah, there's no approval. You just everybody in the world can get in. In fact, when we did it last year, when we had 3,000 attendees, there were people from every continent participating, which is very cool.
Mhm. Sounds good. One last general question is people are wondering about the data security on Galaxy platform, so particular for unpublished data. So can you address that question?
Yeah, so the default for Galaxy is that any data you load in is yours and yours alone. Nobody else can see it, including people who work in the Galaxy project. And that is true. I can tell you now, again, since you're plant biologist, it probably doesn't matter to you that much. I can tell you that if you're working with human data, I don't think it meets whatever the requirements are for working with human data. I can't remember what the deal is. It's my luck that for most of my life I've been working with model organisms, so I've never had to worry about that sort of thing. But for in terms of security for unpublished data, I have absolutely no concerns about it. And when you share data, you can also share things, like I said, you can share histories, which will mean you'd share the data that goes along with it, either with other people or with the world. And so if you share it with other people, again, it is going to be shared just with them. It wouldn't be available any other way. So
Okay.
it's quite secure.
Yeah, wonderful. So as I said before, we have some more advanced questions, maybe that people want to explore more into Galaxy platform. So for that, I would recommend you reach out to Galaxy team directly to answer this on a case by case basis.
That would be fine.
So yeah, so with that, we want to conclude our seminar today. We thank our panelist, Dr. Scott Cain, to give a wonderful presentation, a lot of information about the Galaxy and a lot of about a lot of hands-on experiences for people to get started. So at the end, I want to mention again our website. If you Google our name, Plant Method Exchange, you can find our website. We are hosting some PDF protocols in there, and more importantly, we are doing some monthly or bi-monthly webinars about different platforms and different methods. So you can find us on our website and also find us on YouTube. We'll send out the recording afterward to the attendees, and so that if you miss some part, you can watch the recording. And with that, thank everyone. Thank you, Scott, again.
One
And
thing I just want to
yeah.
say before you head out. One suggestion is when you save all these questions that are in the Q&A and send them to me, and I will add as comments to the YouTube video when it comes out. I will add as
Yeah.
comments answers to the questions.
Yeah, that's wonderful. Yeah, amazing. We'll do that.
Okay, sounds good.
Okay.
All right, thank you.
Thank you.